I'm Dr. Ganesan, Assistant Professor, Department of Genetic Engineering, SRM Institute of Science and Technology, Kattangulattur. In this video, I'll be giving you a brief introduction about chromatin immunoprecipitation technique, in short ChIP. ChIP is a technique designed to analyze interaction between proteins and DNA. It allows researchers to identify which proteins are bound to specific DNA regions and helps in understanding gene regulation. ChIP is widely used in research to study histone modification and to study transcription factors and to study gene regulation. The principle behind ChIP is antibody-based recognition. It involves 4 major steps: cross-linking, chromatin sharing, immunoprecipitation, DNA isolation. In cross-linking, the DNA binding proteins are fixed to the DNA using chemicals like formaldehyde. In chromatin sharing, the DNA is fragmented into small pieces using sonication or enzymatic digestion. The third step, that is immunoprecipitation, an antibody is used to selectively recognize and bind the protein of interest. The last step, the DNA is purified from the protein-DNA complex and analyzed using techniques like PCR, qPCR, microarrays, or sequencing. The amplification levels in qRT-PCR help determine the binding enrichment of the protein of interest at specific genomic location. Higher enrichment means stronger binding of the protein to the DNA region. Lower or no amplification suggests that there is no interaction at the genomic location. ChIP can be performed using 2 different approaches based on whether a cross-linking agent is used. The first approach, native ChIP. No cross-linking agent is used. That means proteins remains in their natural state. The chromatin is directly isolated from cell nuclei. Nuclease enzymes are used to digest chromatin into smaller fragments. In the second approach, chemical cross-linking agents such as formaldehyde used, which stabilizes protein-DNA interaction, and chromatin fragmentation is achieved via sonication or nuclease digestion. Advantages of native ChIP: better antibody recognition because proteins remain unfixed and their natural conformation. Histone proteins are abundant, meaning PCR may not be required for detection. Advantages of cross-linked ChIP: it can detect both histone and non-histone proteins, making it useful for studying transcription factor. Requires less starting material, making it efficient for small-scale sample. Prevents protein loss, which allows the detection of transient or weak protein DNA interaction. Modified ChIP techniques are ChIP on chip, ChIP-seq, native ChIP, ChIP-exo, ChIP-PET, iChIP, nChIP, RIP-ChIP. The limitations of ChIP techniques: they yield low signals, making it hard to draw clear conclusion. If the fragments are too large, it becomes difficult to pinpoint the exact protein binding site. The cross-linking can trap proteins that are near DNA but have no real functional significance. Proteins that bind only for a short time may not be effectively captured. Some proteins interact in large complexes and this can hide the binding site needed for antibody recognition. The success of ChIP heavily depends on the quality and specificity of the antibody used. Thank you for listening.